and data sharing. I have already introduced myself, so feel free to ask me any questions. My name is Emma. You can just call me Emma. As I mentioned, we're at SFU. We're located in Vancouver. In today's session, hopefully by the end of this workshop, you will better understand how variable contextual data, both in terms of its content and its structure, can impact pathogen genomics analyses. You'll understand and be able to describe different types of data standards and how they contribute to data quality, as well as to interoperability between data sets and systems. You will understand and be able to differentiate ontologies from data standards and from the tools that implement the data standards. And you'll be able to describe examples of how they're used for data curation. You'll know the strengths and weaknesses of different public repositories, and you'll be aware of recommended attributes and principles for data sharing platforms. And finally, you'll also be aware of data sharing stewardship principles and different kinds of considerations, such as practical, ethical, and privacy considerations as well. Okay, so if you've taken the MIG course right before this one, or if you're just involved in pathogen genomics at all, you're probably already very aware that pathogen genomics has a wide array of applications. And it's implemented using different techniques, different samples, all with regards to answering a very wide variety of different questions. And the answers to these questions, the information that you derive from these pathogen genomics analyses can be used to as sources of information for innovation. So, answering basic research questions, but it can also be used for informing very practical actions. So, for example, outbreak investigations. But this information also has a lot of impact and is used to inform decisions for a lot of other things as well. So these outcomes can be used to influence policies and laws, the shape of Canadian, but also global infrastructure, computing infrastructure, for example. It can be used to determine who's getting hired, who's getting fired. It can influence international relations, and it can impact funding for a lot of different things. So having high quality pathogen genomics information and trusting that data and how it was produced is really critical for informing a whole slew of different decisions. So when we talk about pathogen genomics, we're of course talking about the sequence data, but we're also talking about the contextual data. And the contextual data is all the information that we need to interpret the sequence data. So it can be things like sample metadata, it can be lab results, clinical or epidemiological data, it can be environmental information, and it also can be methods and metrics that give us confidence in the sequence data that we're using. And the contextual data can help inform a lot of different things. For example, it can help us characterize lineages or sequence types or clusters. It can help us identify variants with clinical significance. It can help us correlate genomics trends with different kinds of outcomes, different kinds of risk factors. It can help us monitor and do quality control, especially when we're comparing results across different labs. It can help us generate hypotheses about the sources of infection or transmission. And as we've already mentioned, critically, it can help inform decision making for different kinds of public health responses, and it can help us monitor the effects of different interventions. So contextual data is really important for pathogen genomics. So data standards can be a great, it can be very necessary for organizing our contextual data. And I'll show you a little bit more of that later in our lab. But essentially why we need data standards is because everyone involved in pathogen genomics, whether it's in public health or whether it's in research labs, everyone is using different tools and they're using different methods. They're using different metrics, different thresholds and criteria. And the contextual data that we capture is usually recorded using either organization-specific data dictionaries or free text. And what ends up happening is that contextual data ends up containing a lot of errors. It can contain jargon or a lot of shorthand notations. And that can contribute to something we call semantic ambiguity. So that's where the meaning of the information isn't necessarily clear across different users. People use a lot of different data structures. I'm sure everybody here has struggled with different dates before, for example, and the information is captured with using different concepts at different levels of granularity, and people use different data structures. So it can kind of be a little bit of a dog's breakfast. And that really impacts how efficiently we can be using the contextual data for our different analyses. And you'll see this later on because in the lab, we're going to be curating data that you're going to need later on in the different modules to do different sorts of analyses. And so data standards are solutions for helping us to make sure that our information is well structured and that it's correct. And data standards, you may already have experienced them before, but in case you haven't, these are sets of recommended controlled vocabulary. They're sets of formats, data structures, and rules for storing, transferring, and expressing information. Usually, a data standard is created by some sort of authoritative source, some sort of uh, body, standards body, and you know, specifications aren't really data standards until there is uptake in the community. So you're looking for something that's created by an authoritative source and that the community actually starts using it. And the variability that we experience in private databases, um, and that can maybe impact our analyses in our own labs, really is exponentially worse when that data is propagated out to different public repositories. So you can see in the slide here, during the pandemic, this is just some examples of data that we found in a public repository. You can see that different fields are being used to capture different kinds of information. There's different formats. Now, if you were to try to integrate these two different records so that you could start to do your analyses, it might take you a few minutes to reconcile the differences here. But if you were downloading hundreds or thousands or tens of thousands of records and you were trying to clean those up and integrate them, it could take you hours, it could take days, honestly, it could take weeks. And in an emergency situation, that's no good. So in a nutshell, you can say that data structure really impacts data function. And without data standards, your cleanup can take a lot of time. It can be very resource intensive. So we want to avoid that. There are different kinds of data standards. There can be standards for individual contextual data records, and that's probably what we're going to be talking about. Well, it is going to be a lot of what we're going to be talking about today, but there are other kinds of standards. There are standards for derived or aggregated data, so the kinds of stuff that might be in reports or dashboards or presentations. There are standards for organizing systems. There are standards such as that prescribe different tricks. There are all
Um,
that are
sorry, I
used
just want to check. Is Emma's voice?
bioinformatics, standardized.
Yeah, it's
Can
cutting
you hear
out.
me? Oh, when did it cut out?
It was just like 30 seconds ago. It was cutting in and out.
Oh, is it better now?
Yes.
Yeah.
Okay. Okay, let me know if it's cutting out again. Apologies for that. So I was just saying that there are data standards that prescribe different metrics and thresholds, and there are also standards that help to standardize the inputs and outputs of different bioinformatics analytical processes. And there are a lot of different standards bodies out there. Some of these you may have heard of, and you may have used some of their standards already. I'm not going to go through all of them, but all this to say that they're in different disciplines, different sectors, different organizations may adhere to the standards of, one standards body over another, and that's fine. Different standards have different strengths and different utility. But just to say there are standards is you're going to be hearing about some particular standards today, but there are quite a few different kinds of standards that are out there. So for those of you who, I think everybody here is involved in research labs, but you may be engaging public health organizations. And whether you are or whether you're not, I think that there are some important considerations when we think about data standards that are used for pathogen genomics, especially in the area of public health. And the first is that in public health organizations, in countries actually that have public health, and that's everybody, there are usually multiple jurisdictions and multiple health authorities that need to interact. In each of these different jurisdictions or these different authorities can have different policies and rules. They can have different data needs because they're asking different kinds of questions. Different health issues are happening in their different regions. They have different chains of command. They have different data access. They have different privacy officers. There can be, that have lawyers and things involved. And there are different kinds of information that are stored in different places. So we need to keep that in mind. You have different bodies that need to interact. And there are also different kinds of sharing. So you can have sharing within an organization because different parts of the organization may play different roles in generating a sequence. There can be people that are collecting samples, and they may or may not be the people that are characterizing the isolates or extracting isolates from the samples. There you can have people who are in charge of doing the sequencing, and they may or not be the same people who are doing the bioinformatics analysis. And then you have people that are making decisions based on those analyses who may not be the same as any of those other individuals. And the nice thing about sequence data is that while it's powerful, while it's generated usually to answer one set of specific questions, it can be reused to answer different kinds of questions. And so you may be keeping that data and sharing it with a different team that's working on another project within your organization as well. So you can be sharing within an organization. You can be sharing outside of your organization with trusted partners, so within a network, and you can also be sharing publicly. And at each of those different kinds of sharing will probably have a different subset of the data. So there's different kinds of sharing that we need to keep in mind. And there's different rules around those different kinds of sharing. In the public health space, auditability is really important. So you need to, and Gary can speak to this, the importance of documenting how your evidence is generated. What were your methods? What were your QC? You need to track chains of custody. You may want to keep accreditation in mind. And this is all because the decisions that you're making that influence the actions and outcomes are critical, and you may need to go to court. And so you need to have all of that evidence documented to back up the actions that are taken. Another thing to keep in mind is that that there are different data needs depending on what's happening. So if you have different pathogens may have different data needs. There might be different data needs in an emergency situation versus general or routine practices. So data needs can change across pathogens and across situations, but also over time. So you need to keep that in mind. And there are also, as we've already mentioned, different privacy, practical, and ethical considerations when it comes to data sharing. And so data standards need to be addressing all of these different things. So out of those considerations and a lot of experience, some best practices and principles for data standards development are emerging. And those, I've highlighted a few of those things on the slide here. Data standards should support data collection globally and also should support interoperability with other standards. Because one thing we're going to say over and over in this class is that there is never one data standard to rule them all. Different standards have their different place. Different bodies are generating different data standards. And so interoperability between standards is really key. Standards should support analyses for the most common pathogen use cases. And standards should also enable and capture potential bias or limitations of the data as well. As we've already said, that's key for public health. Data standards should also support different kinds of data sharing. And it's really key that a specification is established as a standard by consensus. So that can be within within a standards body or across the community. And it's really critical that the group of experts that are generating or involved in that consensus generation represent different countries and are from different settings as well. So you're really getting that input from a lot of different settings. Some of the different best practices, technical best practices, are that standards should be in a format that involves and that are open source and involves a common formal language. I don't mean English, I mean some sort of computing language. They should be machine readable. They should use best practices for semantics, creating new vocabulary. And you should try to future-proof your data standard by considering use cases beyond your immediate scope. So if you create a data standard for enterics, if you want to be expanding that data standard to other pathogens later on, you should be kind of keeping that in mind. And this is all so that your data is understandable by humans and computers. The language that you're using, the vocabulary that you're using is well defined. That the vocabulary that you're using is as universal as possible. So you're sort of, as much as possible avoiding organization or sector specific jargon. And this all makes all of your data easier to index, catalog, and compare across studies and different surveillance initiatives. So at CIDGO, the Center for Infectious Disease Genomics and One Health, where I work, we've created a modular interoperable framework for data standards design. And this is so all of the standards that we make are interoperable, they're consistent, and they're more easily developed. And when I say that they're modular, I mean that all of the vocabulary, the fields and terms that are similar are grouped together thematically. So all the different database identifiers that you need to track, they're all in one module. The sample collection and processing information, that's in another module. All your sequencing information, that's in another module, and so on. And this aligns with an ISO standard for whole genome sequencing that came out a few years ago. And all of the vocabulary that we use to populate the modules are sourced from community-based ontologies, and we're going to get into what those are in just a second. But this modular framework is really great because they're almost like the modules are like cassettes that you can pop in and out. This makes it much easier to use the framework to design a standard for pretty much any pathogen or any target. You can just add or subtract the modules that you need depending on the data needs of your use case, or you can enrich or deplete any particular module for fields and terms. And just to give you an idea of what that looks like, I've aligned two different standards that we've created. One for mpox. So if you were using clinical samples to do mpox surveillance, that's the blue boxes or the boxes on the top line in the figure at the bottom of the slide. Below that in red are the boxes that show you the modules of a wastewater specification that we created. And you can see that a lot of those boxes line up, indicating that the same modules are in the two standards. Where they differ, I mean, because the mpox standard is designed for clinical samples, it has a lot more host information. And in the wastewater standard, there's a module for environmental measurements and conditions. So that's the main differences there. But what this is showing, what this is indicating is that if you had two different labs that were using different strategies for surveillance of mpox, the first lab was using clinical samples. The second lab was using wastewater as a tool for surveillance. The data from those two different labs surveying the same pathogen, but using different strategies, would be much more integratable using two compatible standards built on this framework than, say, if you were using two completely different standards or no standards at all. So, this framework, as I mentioned, helps to generate interoperable data standards, but really something that's crucial for data standards is packages that help implement them. So a standard is only as good as its implementation. And so with all of our standards are supported by packages that contain tooling and different kinds of reference material. They're all version controlled and publicly available. And there are mechanisms for encouraging communication between developers, standards developers, and standards users. And that's because, as we mentioned, data needs can change over time. And so your standards need to evolve with those data needs. So I wanted to take a little bit of time to get into ontologies a little bit, since we mentioned them on the previous slide. So if you're not familiar, ontologies are basically sets of controlled vocabulary that are arranged in a hierarchy. And all of the vocabulary is linked together using logical relationships. So you can see I have a little cartoon on the right-hand side here. This is a mock beer ontology, one of my favorite subject matters. And you can see that all of the different types of beer are arranged with beer at the top and very specific types of beer at the bottom. And they're all linked using the ISA relationship. So a Pilsner is a type of light beer. But you can see on the right-hand side, there are other kinds of relationships that you could use to link beers to different kinds of information, such as ingredients or their qualities, whether they're light beers or dark beers, who produced them, and so on. So an ontology is much richer and much more complex than just a list of vocabulary. The nice thing about ontologies is that they're designed to be as universal as possible. So not just a sector specific or an organization specific meaning of a term, but if that ever needs to happen, sometimes that does, the meanings of different terms are disambiguated with identifiers. So, for example, if you had the word Corona, and Corona being a type of beer, that could be have a beer ontology identifier. But a Corona, of course, is also the light that surrounds um, a sun. And so that's a completely different kind of Corona than a Corona beer. And so the meanings of those two different words can be disambiguated with identifiers. Ontologies also can handle different labels for things. So synonyms, and this really helps with, so the same word like a garbanzo bean would be a synonym for a chickpea. So you can have different organizations that have different preferences in terms of the vocabulary they use. Ontologies can handle synonyms, and that really helps with creating interoperability between organizations. The way you build ontologies, there are whole communities of practice around how these things get developed. And in the community of practice that we're involved in, the Obo Foundry, they have principles and practices that help encourage scientists in the community to be creating interoperable. You're using the same rules, the same principles and practices, so everyone's ontologies are consistent across different domains of knowledge. There's also different registries and portals so that you can more easily find ontologies that already exist that are of interest to you. And there's different languages and tools for developing ontologies and for deploying them and implementing them. And using ontologies is a really nice way to make data fair. So you probably heard of this before, but FAIR is a set of practices, and it stands for findable, accessible, interoperable, and reusable. So the beer cartoon was a very simplified way of looking at ontologies. This slide is just to show you how there are how different tools and portals are used in the ontology development and development process, but also in how you interact with them. So you can see on the top left-hand side, there's a little snippet from a tool called Protege, and this is a very widely used ontology development and editing tool. You can see that you can be creating your terms in Protege here. On the lower left-hand side of the screen, there's another snippet of Protege. This is where you can move from generating individual new entries or individual term entries and how they start to fit into hierarchies that you can build. You can also start to build in the logical relationships between the terms in Protege as well. And then on the right-hand side, this is Ontobe. And this is an ontology lookup service that just contains Obo Foundry-based ontologies. And the Obo Foundry has hundreds of different ontologies that are available that in theory are all interoperable and all consistent. And so, yeah, as this was just to give you the idea of the a little bit more of a realistic view of what ontologies look like in the tools that can be used to generate them and use them. So one example of an OboFoundry ontology is called the Genomic Epidemiology ontology, or otherwise known as GenEpio. So for transparency reasons, this is our one of our ontologies. But ontology is very useful for building data standards for genomic epidemiology and pathogen genomics. And when we start to delve into some of the data standards that we're going to use in the lab part of this, you'll encounter a lot of vocabulary from GenEpio. So GenEpio covers a vocabulary describing different environments, anatomy, that you may be encountering in different sampling strategies. It also includes vocabulary for different kinds of taxonomy, assays, methods, describing all different kinds of things involved in pathogen genomics. And you can see, I just popped some examples of terms at the bottom of the slide here so you can see what the terms would look like in a from when you encounter them in a data standard. So you can see the first part of the term is the label, so the thing itself. And then in the square brackets, you can see the ontology identifier. And the first part of the identifier is the prefix for the ontology. So you can see GenEpio. You know there that this term is sourced from GenEpio. Now, how you can put ontologies to work to start annotating or curating data. This is an example from some work that we did a couple of years ago. We were working with researchers in the child study. So this is all about looking at, this is a very large longitudinal study looking at how asthma and allergy develops in children. And there's a whole bunch of data that gets collected around the children's environment, their health, the medications that they're given over a 5-year period. A lot of samples are taken from the kids. And so you can understand that in such a large study, if things are not well curated, things can get very messy very quickly. And so one of the activities we did was to curate some data about the medications that the children had been given. And this information was captured by the parents of the children. And so they were tasked with recording the different medications that the kids received. And the parents are not professional data curators, so they're writing things down with different brand names, whatever they could remember. Oh, the kid got an antibiotic, I don't remember what it was, or so it made it very difficult to identify trends in the data when the data was so highly variable. And there was also spelling errors and things like that in there as well. So what we were tasked to do was to identify the medicinal component, the active ingredient of the medications that the kids were given, and then to standardize that. And we were able to do that using ChEBI, which is another OboFoundry ontology around chemicals. And so this really helped the researchers identify trends associating different drug exposures with different cases of asthma and allergy development in children. Another type of ontology is called the food ontology or foodon. Again, this is another one of our ontologies. This has tons and tons of terms describing food products, feed, as well as the different kinds of sources of food and different kinds of food processes. Again, it's built on an interoperable architecture that's prescribed by the OboFoundry. But all of this vocabulary is really organized by facets. And you can see that what we mean by facets in the picture on the right-hand side of the slide. So food products can be organized according to food product type. They can be characterized according to the kinds of consumer groups. Who are the people that are meant to be eating these foods? Is it children? Is it adults? Is it people on specific diets, for example? You can organize or characterize different food products according to the different kinds of transformation processes they undergo, and so on. Now, the nice thing about food on is that with all its facets is that it can be used to break down complex descriptions of food into standardized and mappable units. So I don't know if you've ever worked with food data, but a lot of times it's captured using free text, and you can get a long string of words to capture and to characterize the food that was eaten. So you can see an example in the slide here: organic mashed carrot baby food jarred. And so to use this data to be able to extract trends can be very difficult. Um, and so what we can do is actually use food on to extract different pieces of information from these kind of long descriptions and to characterize that information or to categorize that information so that it's much more usable. So you can see the facets here: label claim, food processing type, food product organismal source, and so on. Each of these are different facets from food on, and we can use each of these different facets to create new data elements. And we can break this down so that you can start to analyze this information, different cuts in different cuts of the data. Now, the nice thing about ontologies is that they are, especially if you're using ontologies built, in a community of practice like the Obo Foundry, is that all of these different ontologies built for different domains of knowledge can all interact. So one of the most important principles in ontology development is reuse. So if there was a term that was created in one ontology and you need that term in your ontology, you reuse that other ontology's vocabulary. So you can see here in the slide a picture that shows the vocabulary that food on is contributing to the community. So you can see in the orange circles that food on is contributing vocabulary in the area of harvested materials, food products, consumer information. But you can also see all the interactions with all of the other ontologies. So you can see that there are ontologies like DOID, Genepio, Fideo, for example, that are also pulling vocabulary from those domains: harvested material, food product, and consumer. So they're taking terms from food on and reusing them. And food on is also pulling, you can see with the lines that are connecting them to other ontologies like Uberon, so that's the anatomy ontology, for example, or Kebi, because there's a lot of chemicals in food. Food on is also pulling information and vocabulary from other ontologies as well. And this creates an ecosystem. And this is really powerful because data that's annotated with Obo Foundry ontologies, different Obo Foundry ontologies, you can start to create knowledge webs. So you can interconnect different data sets from different studies to be able to do more interesting things, more complex querying. I know people here where someone was mentioning that they're involved with AI. All of that is empowered by using these interconnected ontologies. Ontologies can also be used to do a lot of different data modeling and building graph databases. So, the example that you can see here, I'm not going to go into this in detail. This is an example of a data modeling exercise. So this is a positive HPV status, a person whose sample tested positive for HPV, and the person was also dealing with a head and neck cancer. So this was a patient who was also associated with an HPV test. And you can see that this gets very complicated here, but this information would be very difficult to work with if you're just using a flat file. So you can see that using ontologies, you can start to integrate all kinds of data from different sources, and you can start to ask a lot more complicated questions. So I'm not going to go into this. Doing this stuff with your data would require a whole other different course. But all this to say that if you are annotating and curating your data using ontology-based data standards, you're setting yourself up and empowering yourself to do a whole lot of new and more exciting downstream analyses. And there's a growing ecosystem of interoperable data standards out there that are based on ontologies. So some that we've built, some that others are building out there in the community. So we have different kinds of contextual data specifications, and you're going to be working with some of these data standards later on in the lab. But there are also, as we mentioned as well, there are other types of standards that empower our bioinformatics analyses. So standardizing inputs and outputs, metrics, for example. There's a really nice tool that was built by the Public Health Alliance for Genomic Epidemiology, and that helps to standardize the outputs of different AMR detection tools. So these different tools can be very variable. They're using different databases. They might be looking for different kinds of resistances. And so it can be really difficult to align the outputs of different tools and to assess maybe, which tool is better for your analyses. And it makes it kind of difficult to share data as well between labs when, the outputs are all variable. So harmonization helps to align all of the different outputs, and it creates a nice report that you can work with as well. So I'm not going to go into all of these different standards, and if you have any questions, feel free to ask. But all this to say, there is a whole library of standards that are out there that can help support your analyses. And just to give you an example, during the pandemic, it was known that because health is under provincial jurisdiction in Canada, that means that each of the provinces gets to say what kinds of data is collected, what are the questions that they're interested in answering, what samples get collected. If we wanted to take all of the information that was being generated by the different provinces and pull that all together into one national database in order to understand different national surveillance priorities, we would need a data standard to be able to harmonize all of that information in one place. And so our lab was really fortunate to work with the Public Health Agency of Canada and a lot of the different provincial public health labs to create a common SARS-CoV-2 contextual data standard, and that was implemented in a tool called the Data Harmonizer that we're going to talk about in just a second. And through the use of the standard and the tooling, that really helped to mobilize and harmonize data from a lot of different sources so that it could go into a national database and be used effectively. Another example of a data standard that has been used, this was created again by the Public Health Alliance for Genomic Epidemiology, and actually a number of us, of the instructors that are on the call today are actually involved in PHAGE, including myself. So this PHAGE developed a data standard for wastewater genomic surveillance. And this standard has a number of different modules. We've discussed this already, actually, when we were comparing this to the MPOX standard. So you can see some of the different, what the different modules are on the left-hand side of the screen. If you want to read more about this, I know someone mentioned that they were interested in wastewater surveillance. If you're interested in learning more about this, and I popped our preprint and our GitHub link at the bottom of the slide. But this standard was piloted and tested in over 15 different countries across 5 different continents for use. And it actually has now been used as a subset of this standard has now been deployed as a new biosample package at NCBI. So this is helping to align data from all the way from local sources, local labs, to global use. So you can see I popped the biosample template on the slide there as well, and there is another compatible ENA biosample package coming soon as well. So that's for international sharing, but the data specification itself can be used in your local labs to help organize and get your data standardize your data for different kinds of wastewater analyses. Okay, so as I said before, a data standard is only as good as its implementation. So when you develop a data standard, you really should be putting it into some sort of tool to enable people to operationalize it. And so in our lab, we've created a tool called the Data Harmonizer. So as I mentioned a few slides ago, this was used during the pandemic to harmonize all of Canada's SARS-CoV-2 data that was being used for national surveillance. So the data harmonizer, you can see here, it's a spreadsheet style text editor. Um, there are different templates in the data harmonizer that implement different standards. So for example, you could see the SARS-CoV-2 standard, it would have its own template. The wastewater standard would have its own template. And there are more, and we'll be working with those in the lab later. You can see there are some curation bells and whistles in here that can help you help speed up your curation. The data harmonizer also validates your data against the standard as well. So it'll help you identify any, if you've misused any drop-down menus or you haven't filled in things that you need to fill in. It's also color coded as well to help you identify which fields are absolutely required, and those are highlighted in yellow, and there's usually only a handful of those per template. But there are a lot more recommended fields, and those are colored in purple, and all the gray fields, those are just optional. And that's because sometimes labs only collect a small amount of information or only are wanting to share a small amount of information, and they're using the data harmonizer as a mechanism to standardize their data for sharing. But other times labs want to standardize a lot of information and keep that for themselves. So the data harmonizer allows you to align your data with a standard in different ways. The other nice thing about the data harmonizer is that it enables automated transformations of your data. So another big thing we do in our lab is we spend a lot of time mapping standardized terms to different downstream formats. We know your data needs to move around. Often it needs to move around. And there are different standards that are used for different purposes out there as well. And so we map all of our standards to as many different standards that we know about, and we encode different exchange patterns, I guess you could call them, different exchange formats in the data harmonizer. So basically, you can enter your data once and press a button, and it'll reformat that data for you and export that. And that's because we don't want you to we want you to spend your time doing analysis and not reformatting your data for uploading to a repository or sharing to a network in a different format. So I've highlighted the data harmonizer, but there are other data curation tools that are out there. I've popped them on the list here. There's probably lots more out there. And we are interested in creating a list of these different curation tools. So if you are creating a curation tool or you're using a new curation tool that you don't see on this list, please let us know. We would love to know about that. But all this to say that there are other tools that are out there. I don't have time to go into them all today, but the data harmonizer is one of those things. So I just wanted to take a minute to differentiate between some of the different resources that we've learned about today. The first is ontologies, and the second is specifications or data standards, and the third is tools. So ontologies are meant to represent things in a universal way, right? We know that we have standardized terms, they're in hierarchies. There are lots of different annotations that can be and information that can be included in ontologies. But these are really sources of standardized terms, right? That's what you can think of them as, sources of standardized terms. Those standardized terms can be used in your data standards. So you've plucked out the bits that pertain to your data and you've put them into a data standard. And this is where your data types, your rules, your patterns, your max mins, all of those kinds of and your formatting, all of those nice things go into a data standard. They don't go into the ontologies. For example, if you have sample data, uh, sample collected date, sample collection date. That can be a field or that can be a term that's in an ontology, but you would specify the format of the date that you want in your data standard, right? Because you can have different date formats, and the ontologies aren't the place to be prescribing those things. So we have ontologies, we have specifications or data standards, and the specifications are implemented in different tools in data management systems. So you can have the same data standard implemented in a lot of different tools. You can have tools that implement a lot of different data standards, but these are all separate things. And the kinds of different tools and data management systems that can implement data standards are things like spreadsheets, lab information management systems, different kinds of databases. They can be your own private databases, they can be public repositories, and other things that can be implementing standards, as well as ontologies, actually, are different kinds of online tools like lookup services. There are different kinds of curation or validation tools, transformation tools, mapping tools, and so on. So the main take-home message here is that ontologies are different than data standards, and those are all different than the tools that implement them. Okay, so now we're going to get into data sharing, and specifically different kinds of public repositories. So submitting data to public repositories is incredibly important for global surveillance. And being able to use other people's data is incredibly important for your own situational awareness. So understanding what's circulating in your own communities and comparing that to what's circulating in communities around the world provides a lot of insights into how pathogens are evolving, how they're moving around, how they're getting into your communities. And we're going to get into why you should share a little bit later. But right now, we're just going to talk about mechanisms for sharing. So there are several different public repositories out there. I'm going to just mention a few in the next few slides. They all of these different public repositories have their different strengths in terms of how easy it is to submit, how easy it is to retrieve information, either the complexity of the data that they hold or the simplicity of the data that they hold. So there are a lot of overlaps between different public repositories, but they often have different requirements when you're submitting. Um, so if you are using if you are using a data standard, often you're going to need to reformat your information before you can submit to any particular repository. Um, and what you end up sharing out of, whatever you've standardized using your data standard, that's up to you, and it can depend on your own institutional data sharing policies. But no matter where you choose to share your data with, if you do choose to share, generally speaking, you're going to need to set up an account. You're going to need to prepare your contextual data and your sequence data. So you're going to have to set up your files, and then you're going to need to submit. And each public repository has its own process. Some of them, you can do programmatically. Other times you have to use a particular user interface. But those generally are the three steps. The first public repository we're going to talk, or set of public repositories that we're going to talk about, is, if you're in this course, you're probably familiar with this already, is the INSDC, so the International Nucleotide Sequence Database Collaboration. And so this is a collaboration between three different nodes. One is located in the US, one is located in the UK, and one is located in Japan. And while you can upload to one of these different nodes, the data is mirrored across all three nodes nightly. That is when governments are functioning as they should. And each of the nodes, while the formats generally overlap quite a bit, they each have their own submission requirements, and they can have their own data structures. But they do reconcile these between the different nodes when they're mirroring the data. Um, and the reason why there are these differences is that each of the different nodes is funded differently, and they have different priorities. But it's an incredible collaboration that is we don't really see anywhere else in the world. So the INSDC has been around for a long time. They host all kinds of data, human data, pathogen data, all kinds of data. And you can set things up there using these things called bioprojects. So you can submit whatever you want to a bioproject. Within the bioprojects, you can have sample records, and these are called biosamples. And then there are different places to be submitting different kinds of data. So, there's a database for raw reads, for example, and then there is a place for assembly or consensus sequences. So there's a really nice structure for submitting data. When we're talking about contextual data, the INSDC has a tremendous library of biosample packages, and these are mostly built off of Genomic Standards Consortium checklists. So I've mentioned a few here that might be of interest to you. I would say, generally speaking, and I'm probably preaching to the crowd, the choir already, you probably already know. Generally speaking, the pathogen packages are probably going to be your best bet if you're thinking of submitting to the INSDC. So the pathogen packages can be clinical or host associated, or you can have an environmental or food package as well. Okay, another public repository I wanted to mention was GISAID. So let me see if I can remember what this stands for. The Global Initiative of Sequences for All Influenza Data. Basically, this was originally started to share influenza data. And it has been incredibly powerful for getting people to share because this is a controlled access repository. So when you go to sign up, when you go to register, you sign an agreement, and there are different rules around data attribution, collaboration, data use, and you have to adhere to these rules. GISAID has expanded to other pathogens besides influenza. So they also accept SARS-CoV-2 data, Mpox data, RSV data, different kinds of arboviruses as well. And there are separate databases within GISAID for each of these different pathogens. You submit your data using a particular template. All of the templates look much the same. So if you get the hang of one, you can easily get the hang of the others. GISAID was a game changer in the pandemic. Many public health labs actually started sharing data when they maybe didn't share data before because uh, of protections that GISAID offers. So you're not supposed to, you can use the data there for public health analyses, but you're not supposed to publish using the data unless you go back to the original submitters and ask them if they want to collaborate. So that it was incredibly powerful during the pandemic, but there are a number, and it's easy to submit there, but there are a number of community concerns around governance and the transparency of their practices. And I've just popped a little link to an article at the bottom of the slide here that sort of touches upon these transparency issues, if you're interested in learning more about that. But as I said, the GISAID was really powerful during the pandemic, and kudos to them for that. So just to contrast this with INSDC, INSDC is completely open access. So you don't have to check in with the data submitters when you want to use their data. You can do whatever you want with it. Whereas with GISAID, there are rules around what you can do. That can be a little bit limiting, but those rules are there for a reason. If you are interested in sharing to either GISAID or any of the INSDC nodes, but you're a little bit, you're unfamiliar with how to do it, Phage has put together protocols for how to submit data and how to choose the right biosample, et cetera. So if you want to pop over to our protocols.io workspaces, you can find those different submission protocols there. The last repository that I wanted to mention was Pathoplexis. So Pathoplexis really is a new kid on the block. And it just went live in 2024, and it's focused on hosting viral data. And you can see the different pathogens that it is focused on the right-hand side of the screen. You can see there's a little bit of overlap with GISAID. Again, you have to register. There are usage restrictions very similar to GISAID. But what's nice about Pathoplexis is that it helps to automate upload to ENA after a year. So it'll do it for you before that if you want to pull the trigger. But you can upload your data for public health use. That can, gives you time, gives you up to a year to publish on that data. And then they will help escort that data into ENA and get that into that INSDC collaboration after 1 year. So that's really helpful. They also version control all their data so that you can track if people have detected some sort of issues and they change their data. That's all transparent. It really encourages the community to make notes about the data as well. So you can write to Pathoplexis and say, hey, I think this collection date's wrong, or there's something going on here. And they will annotate the data and so that the community can learn about those issues. Pathoplexis implements data standards. Well, say they're phage data standards, but if you're using a CIDGO data standard, which you will be working with later on in the lab, their metadata is compatible with those data standards. And it's very easy to upload to Pathoplexis. And the most important things about for the Pathoplexis group is that all of their practices are highly transparent, as much as possible. The meeting minutes from their meetings are all, online, for example. All of the decisions that they make are documented, and everyone's access, there's a set of rules for getting access to things, and that's all very transparent. So if you're interested in sharing viral data, this is a great place to do so. They do have far fewer sequences at the moment because they're so brand new compared to either GISAID or the INSDC, but their library of pathogens continues to grow and people are submitting to them all the time. Pathoplexis data was involved in different Ebola and Mpox outbreaks, resolving those outbreaks in the last year. So kudos to Pathoplexis for doing so much in such a short amount of time. So recently there were some documents that came out of the WHO and the IPSN, so the International Pathogen Surveillance Network, which is a new organization coming out of the WHO. So there are two documents you might be interested in reading up on if you're evaluating different public repositories, or you are thinking of using other data sharing platforms, or perhaps if you are even considering developing a data sharing platform for your organization. So these principles and attributes that are described in these two documents really emphasize the importance of fair data and making your data as reusable as possible. The principles and attributes promote the promptness and openness and usefulness of data. So you make your data useful by annotating it with methods very well, highlighting any limitations, making sure it's as detailed as possible. And they also emphasize, robust metadata, clear access protocols to your data sharing platform, and trustworthiness. So making sure that all of your governance is transparent. It's clear who's funding your platform, et cetera. And they emphasize ease of use as well. So those are a couple of documents that you might want to have a read of. Okay, why should you share data? Now, as I said before, you do not have to share data if you don't want to. That's completely up to you, up to your lab, up to your institution, and you share according to your institution's policies. But I would like to make a plea for why you should share data. And as I previously mentioned, sharing your data and being able to make use of other people's data is critical for situational awareness. The lack of data creates blind spots. It can be blind spots within your own country, or it can be blind spots in other places in the world, and that's not great. It's also important to share because you want to make sure, I've said viruses here, but it can be whatever, pathogen. You want to make sure the sequences of the pathogens that are circulating in your community are involved in the development of different diagnostics and therapeutics. You want to make sure those things are effective against what's circulating in your community. So that is a big benefit to make sure everyone's aware of what pathogens you've got. By sharing data, it gives you a voice in global decision making. And it may not feel like it if you're a researcher in a lab, but sharing is very powerful. The more data you share, the more leverage you have. Data sharing has also been put forward as a key component in a human rights framework that was developed by GA4GH. That's the Global Alliance for Genomics and Health. So they've really looked at the Declaration of Human Rights, and there are certain key points in there that could be argued to pertain to pathogen genomics data. And so, sharing that data is a human right. And then also, of course, just being a good data citizen. So sharing data is important, and I hope that you do it. Data stewardship. So we've discussed places where you can share your data. We've discussed reasons why you might want to share data. If you do want to share data, there are some things that you should be keeping in mind. And to make sure that your data is safe, to make sure that your data and your practices are trusted, and to make sure that your data is accessible and usable. So the first thing that you should be keeping in mind is protecting privacy. So whether you're a researcher or whether you are, a public health scientist, I should say an academic researcher or a public health scientist, you want to make sure that you protect privacy of your data. Now, privacy can be pertaining to, say, human information, human records, human health records, but if you're doing something like monitoring pathogens in particular environments, or you're monitoring foodborne pathogens in specific food, you have to keep reputational damage for different companies, different regions in mind as well, because there can be consequences to sharing specific information about different environments or different products. So you want to make sure that the privacy of your data is protected, because this all really boils down to public trust, right? And the loss of trust has consequences. I come at this more from a public health perspective. If the public loses trust, then they won't get tested, they won't comply with public health actions, they won't get treated, and that can be pretty diabolical. But of course, we've also talked about reputational damage as well. So, if there's a outbreak in a particular kind of food, and you do have to do a recall, that can be very damaging to a company's reputation and their bottom line. So always keep that in mind. So data usually needs to be de-identified, so no names and addresses. Be careful of small case numbers. So if you have only a few sequences and they're in a particular geographical location, or you're combining information with different kinds of fields that can potentially become identifiable. So just keep that in mind. It's really important to track identifiers, especially if we're swapping isolates between different labs or different organizations. We don't want to be sequencing the same thing a million times. But it's also good practice to not put identifiable information in the IDs themselves. But sometimes personal health IDs or sample IDs can be considered identifiable information. So you have to be careful when you're sharing those things as well. If you're sharing like a Toronto lab ID, people know what the convention is for a Toronto lab ID. You might not put Toronto in the record, but they can tell from the ID. So just have a think about that. And if you are involved in an institution that has a privacy office, you should consult them before sharing any data. But just know that they're probably going to be pretty risk averse, but they're going to know more about your institutional policies and perhaps jurisdictional and national legislation that you should be keeping in mind. I think everybody here is in research, so generally speaking, it should be up to you and your PIs who will be the data stewards. In terms of security and quality, always put as much provenance and methods, and that includes any kind of microbiological methods, sampling methods, not just your sequencing and your bioinformatics methods, but all of those things, your reference databases, the tools you're using, the versions, all of those things are incredibly important. And it's all part of the evidence, right? It's all part of what you need to be recording. Those things are all important for attribution and audibility and reproducibility and accountability as well. Your contextual data, so all that nice clinical environmental information, may require storage somewhere with higher security than the actual sequence data itself. Because you can do things with sequence data without much metadata, but it's really the contextual data that's got the spicy stuff in it. And in terms of quality, if you detect any errors, whether it's in the sequence data or in the contextual data, it's good practice to correct those as quickly as possible. And if you've shared that data within your network or within public repositories, don't forget to correct those records as well. Okay, I'm almost done. I have a bit of a pyramid here of the likelihood of being able to share different kinds of contextual data. So at the start of the slide here, you can see types of information that are kind of bolded and in big letters. This is usually the least controversial types of information, so you should be able to share this. So things like geographical location of sample collection at the country level is usually okay, sometimes state, province. As you start to get more granular in your geographical locations, this starts to to not be so cool to share. You should be able to share sample collection dates, hopefully to the day. Those are the most useful, and we'll see that later on in some of the downstream modules. Attribution, so who collected a sample, who sequenced it, all of that information is really important to share, especially if people need to get in touch with you, if they want to use your data or they want to find out more information. Also, acknowledging the different contributions of different folks is really important to include in a record as well. And as we mentioned before, methods. The instrument, consensus sequence software, again, that assembly software, some metrics about quality. All of those things shouldn't be too difficult to share. You should be allowed to share them, but again, everybody's labs are different. The next level of shareability is data that is a little bit more informative. So the stuff at the top of the slide, this is for very basic surveillance. The stuff that's in the next section is a little bit more informative, but is usually a little bit more tricky to share. Things like sampling strategy. So, why are you selecting different things? Why are you what areas you were sampling. Were the samples involved in an outbreak? That sort of thing can be a little bit tricky to share. Information about your hosts, particularly age, gender, other types of information as well, maybe where they work, the kinds of exposures. Sample types and sampling methods. Usually you should be able to share these things, but again, some people may not even have access to that information. As we've discussed, there are different roles, and the lab that's doing the sequencing and maybe the submitting may not have access to, the database that has all the detailed information about the sampling. Also, different kinds of quality or diagnostic indicators, like if you've done an ECT, PCR, and you've got CT values, those are really useful to share. But you may or may not be able to share those things. And the last category of information is probably the most informative, but also often very difficult to share, again, because of access or because of privacy concerns. So, information about vaccination or other types of treatments or therapeutics that have been applied, exposures, travel history, hospitalization outcomes, that all that information can be kind of hard to get, but and also kind of hard to share. One last, as I kind of close up the lecture, I just wanted to make a little bit of a plea for the formalization of curation in different kinds of pathogen genomics projects or programs. Data curation, as you're going to see, as we're going to practice in the lab, is really critical to making sure that you have good quality data and that you have good quality information for decision making. But oftentimes, curation is something that people do at like 5 to 5 on a Friday. People aren't aware of what data standards are already out there. They're not aware that using data standards is a good practice. And so, data curation and contextual data are often treated as second-class citizens compared to the sequence data. But I would like to assert that curation is incredibly important, and there should be dedicated personnel for implementing standards, and that should be embedded in every project and every program. And there should be resources, especially funding, included in budgeting at the outset. And curation and data standards should be integrated into all your experimental planning right at the start in your experimental design. So these things shouldn't be rushed and they shouldn't be an afterthought. So everything that you're learning here today is really important for all of the other downstream activities that you're going to be learning about in the rest of the course. I also wanted to say that data curation and the use of standards is, we're learning about it here for the purposes of learning pathogen genomics, but actually data curation is incredibly important for anything. We live in a data-centric world. So whether you go on and you want to work for the government or you want to open your own, music shop or something like that, we live in a data-centric world. Having good data curation skills will help you with pretty much any job that you want to apply for. So data curation and using data standards, this is a data science. And so, like I say, incredibly useful for all kinds of careers. Okay, so second last slide. The take-home messages for today is that data curation and standards are part of data quality frameworks. Ontology-based data standards increase interoperability and reusability of data sets. Data management tools like the data harmonizer, but there are other ones that are out there, help to operationalize data standards. Data sharing is important for situational awareness, for decision making, but also for innovation, building new tools, understanding how the world works. Data stewardship involves considerations important for data sharing, like the accuracy of your data, the timeliness of sharing, protecting privacy and making sure your data is secure, making sure your practices are trustworthy, and that everything is done in an equitable way. Everyone is acknowledged. These are all very important things to keep in mind, and that there are open and access controlled public repositories out there, and they have different advantages, and they also have different submission formats. So basically, just reinforce that that data curation is really important to making good decisions and for future-proofing your data so that you can use it to answer different kinds of questions, and you can really maximize investments in sequencing and generating that data. Okay, this is my last slide. Thank you so much for listening. I'm going to stop now and see if anybody has any questions. If you want to get a hold of me, my email is there. If you want to learn more about our standards, you can pop over to CIDGO. And with that, I will stop.